So in this video, I'm going to talk about the exact steps of meiosis. And so I think this is a helpful diagram to essentially compare the two, mitosis and meiosis. And remember, meiosis is to make gametes. And so meiosis is split up into two steps, meiosis I and meiosis II. And so meiosis I is the reduction division, in which the chromosome number is cut in half. As you can see right here, meiosis I, it goes from 2n to 1n. And meiosis II, people generally like to say that this is pretty similar to mitosis. So similar to mitosis. And meiosis II, in the end, produces 4 haploid cells. And so now let's talk about meiosis I. Let me clear this. So meiosis I obviously still starts with prophase, but this time it's numbered, so it's prophase I, and meiosis II has a prophase II, but let's talk about this first. So what happens in prophase I? Well, so it's important to know that here we have 2 sister chromatids, but this together, this whole thing is called a chromosome. A chromosome, remember, made up of 2 identical sister chromatids. So let's say we'll use black for your dad's chromosomes and red for the mom. And so remember, these chromosomes have duplicated. So normally there would only be 1 chromatid. And so these are homologous. Let's just pretend these are homologous chromosomes, and they pair up. So they essentially pair up, and it's called synapsis. Synapsis. And then when they're all lined up, so they're lined up, let's pretend that's all lined up, they're next to each other. And this whole structure is called a tetrad. And so what can occur is crossing over can occur, which is essentially just exchanging different parts. So if crossing over occurs, the chromosome may look something like this. So originally you had something like this. However, crossing over is essentially just exchanging genetic material. So what may happen is that part of the mom's chromosome becomes attached to the dad's. It essentially like switches place. And so here are the new chromosomes that result from crossing over. And crossing over occurs at the chiasmata. And remember, crossing over involves exchange between non-sister chromatids, so the red and the black, essentially, because it makes no sense to exchange red and red because they're exact copies of each other. And so these new DNAs, or chromosomes, are called recombinants. And one thing I forgot to mention, it's kind of important, cohesins. They hold the homologs together, so they essentially help make up the whole tetrad. And so one final thing, prophase I is the longest and actually is about 90% of the whole meiosis process. And the number of crossovers that occur down here is completely random and is rather unpredictable. And so now let's talk about metaphase. So I'm going to clear this. And so metaphase is pretty straightforward. The homologous chromosomes, let's say we still had the recombinants there. The homologous chromosomes line up on the plate, the imaginary metaphase plate, and that's pretty much it. I mean, it's pretty similar to mitosis. Really, meiosis and mitosis are pretty similar, but the prophase is the big difference between them. So anyway, this happens in my metaphase 1, and now anaphase. Let me clear this. So for this, I'm just going to use a diagram down here. The synapsis breaks up, so essentially the four chromatids separate, and so what happens is they move to opposite poles right there. And it's important to remember that the chromosomes still have two chromatids attached to them. Like, they're still made up of two chromatids. And now telophase. Let's talk about telophase. And so in telophase 1, the daughter cells have one initially duplicate chromosome from each homologous pair. And so remember, that means that this is 1N and this is 1N, which means that they are haploid. And so after telophase, cytokinesis can occur, or it could go to interkinesis. And what that is essentially it's similar to interphase. No DNA replication occurs. And so that's pretty important. And then now meiosis 2. Let me clear this. So meiosis 2. Meiosis 2 is actually indistinguishable from mitosis of two haploid cells. So essentially you could just think of it as pretty much just mitosis in prophase, metaphase, anaphase, telophase. And I have a previous video on that, on mitosis. But meiosis 2, most people just consider it pretty much the same as mitosis. But the key thing is you could distinguish meiosis 2 from mitosis by looking at the number of chromosomes. Because remember, meiosis 2 is 1N and mitosis is 2N.